Today I'm going to start talking about T cells, which is going to lead us into a discussion, or the beginning of our discussion, of something called MHC, which we're going to spend a bit of time on. I'm right now leaning I'm going to say I don't think we're going to do the paper next Friday. I think we'll probably push it to the Monday after. I will keep you updated on that. And when we're going to do that next MHC related paper. What I want you to kind of think about, and what I always like thinking about when we are starting our T cells and MHC discussion, is sort of this idea of just the fact that we've talked about a lot of immune mechanisms at this point. We've seen a lot of immune system, and so we might have the question of, well, why the heck do we need more? I feel pretty protected with my antibodies and my B cells and my macrophages and my complement and all of that stuff. What do I need more stuff for? And so I want to show you a couple of different scenarios where immune responses that we've talked about thus far wouldn't work, so that we can start to see why we still need this other portion of our immune response, which is the T cell response. And so one of the things that we've talked about is phagocytosis by innate immune cells, like phagocytes. And so you can see an example of phagocytosis happening here. And I will admit that right now I'm using this to summarize the entire innate immune system, which is a little bit of an oversummary, but that's okay. We'll go with it. It turns out that there are some microorganisms, particularly some bacteria, that have evolved ways to inhibit dying by phagocytosis. So this shows sort of 3 examples of that. So after we phagocytose some bacteria, and have them in the phagosome, as is shown here, there are actually some who have evolved to puncture the phagosome and get out of it so that they don't get destroyed in the phagosome. Listeria monocytogenes is the most famous example of this. And there are others that either are able to modify the biology of the phagosome to prevent it from fusing with a lysosome, or otherwise just make it a nice habitable environment. Things like Salmonella typhimurium are some good examples of this. And thus, instead of the phagosome being a organelle that is going to result in the degradation of that bacteria, the phagosome is now a nice little home for that organism, where it's protected from other things getting lots of nutrients, all sorts of stuff like that. And so this is a situation that we can't do anything about with the immune responses that we've seen thus far. We're out of luck with this. Similarly, we can think a little bit about antibodies and ways that antibodies can be useful and ways that antibodies are less useful. And so here you can see an example of an antibody neutralizing a virus and blocking that virus from getting into a cell to replicate within that cell. And that's fantastic. However, if we think about a full microbe, like a virus, like HIV, as is shown here, that antibody is going to be able to detect these surface antigens, like the yellow one and maybe the red one and things like that. But the antibody is not going to be able to bind to things that are inside the capsid of the virus. The antibody can't go into the middle of the virus. The antibody can't go through the virus's lipid bilayer in cases like this one where the virus has a lipid bilayer. And so you can kind of think about it as the antibodies are limited to really looking at the outside of the microbe to determine if that microbe is good or bad. And you can, again, sort of, if we make a parallel or make an analogy here between this and the TSA, this would be like the TSA looking at your suitcase and being like, that looks like a nice suitcase. You can take it on the plane. And not actually looking at anything inside. And so it could be really easy for a microbe to shift parts of the outside of the microbe and keep the inside completely the same and conserved and get away from antibodies, because antibodies are only recognizing the outside parts of the microbe and none of the inside parts of the microbe. Similarly, if we think about a viral replication cycle and a bunch of steps that are required for a virus to reproduce, antibodies are going to be really good at neutralizing and acting on that virus when it's outside of the cell. But for the majority of the steps of the virus's life cycle, that virus is inside the cell. And antibodies aren't going to be able to go inside our cells and get rid of different microbes. And so most of the time, the antibodies are not going to be very helpful once we have any of these microbes inside of our cells. And so what I hope you notice in all of these examples is that the problem that we kind of have in general is the insides of stuff. We might have a bacteria hiding inside one of our phagocytic cells. We might have a virus reproducing inside of one of our cells. And we might want to look at the insides of a pathogen and not the outsides of a pathogen. And you can kind of think of that as a general theme of what we're thinking about here. And in order to deal with all of these cases, we have to have this other arm of the adaptive immune response. And you can think about it that this other arm is the arm that's helping us look at the insides of cells. So we're looking at what kind of microbe is outside of our cells. We're looking at, are there microbes inside of our cells that are potentially able to cause us harm? So that would also potentially help us look at what's going on inside of a phagocytic cell to see if something's trying to live there. It also might let us look at what's happening inside of a microbe as opposed to just outside. And so all of these things are dealt with by this other part of the adaptive immune response, which is the T cell arm. And so you can see T cells that are sort of shown here in purple and blue. They are lymphoid cells. They're lymphocytes. So they kind of come from a similar progenitor to our B cells, but they go through a different developmental process. You can also notice that I've got a lot of branches shown here of a lot of different kinds of T cells, which we'll be getting to eventually. And so the T cells are really what are going to solve some of the problems that I've set up for you. If I was going to make a one-sentence definition for B cells, before my sister yelled at me because she doesn't like it, but whatever. She's not here. I would say B cells make antibodies, right? They're antibody-producing cells, and that's how they deal with microbes. It's harder to make a one-sentence definition for T cells. There is a one-sentence definition out there, and it's a terrible definition. And that one-sentence definition is that T cells interact with other types of cells and make them change. Which is that's not useful, not helpful to anyone. The reason why we have such a bad definition that we use if we were going to have an all T cell definition is because we have so many different kinds of T cells, and they each do slightly different things. And so very often when we talk about T cells, we're going to have to specify which type of T cell we are talking about. broadly, we're going to distinguish those T cells into a type that are known as CD8 T cells. You can see CD8 T cell both at the top here and at the bottom. I know this is going to be a shock, but the way we distinguish CD8 T cells is we look for this protein on their surface called CD8. Alternatively, we have another type of T cell that are called CD4 T cells. You can see CD4 T cell is listed at the top, also here at the bottom. Again, shockingly, we distinguish CD4 T cells because they have this surface protein called CD4. CD8 T cells specifically act by interacting with other cells that are infected with things and killing those cells. So sometimes we talk about CD8 T cells as the killer cells. So you can see CD8 T cell in purple interacting with this infected cell. It's infected because it's got red dots in it. And then that cell getting killed. While we can see our CD4 T cells here and here, they're blue, and they are making cytokines that might do things like change the effectiveness of macrophages, change the effectiveness of B cells, or they can actually influence lots of other cells. The really key thing about those CD4 T cells is really their ability to produce a lot of cytokines. And we often will refer to them as the helper T cells because they help other cells function better. And so you can see that overall, in both cases, our T cell is interacting with some other cell and doing something to it, but though something is really different between a CD4 T cell and a CD8 T cell. In order for our T cell to interact with whatever cell type it is interacting with and induce whatever change it is doing, it needs to have a receptor on its surface. Technically, we're seeing this, or this receptor. And again, because immunologists are so creative, this receptor is called the T cell receptor. Just like if you remember, the B cell receptor was on B cells. The T cell receptor is shown here on the right. What you can notice is that the T cell receptor has two protein chains, one shown in yellow, one shown in green. And in fact, as you will see later after break, one of them is a light chain and one of them is a heavy chain. Oh my gosh, the one that's yellow is a light chain, the one that's green is a heavy chain, just like that you saw in the antibody. You can see there's a disulfide bond between them. You can see there's a super short, there's a transmembrane domain and a super short cytoplasmic tail. You can see that this has some immunoglobulin domains. The outer one on both chains is the variable region. The inner one is the constant region. You can see our antigen binding site that's made up of the two chains together with like three little loopy parts. Realistically, a T cell receptor structurally is really similar to just an FAB of an antibody with a transmembrane domain added to it. So you can think of the T cell receptor, at least structure-wise, as being really similar to what you're seeing here with this FAB portion. So one of the most important things or experiments that have been done to understand T cells is this experiment that I'm going to start telling you about. And really what this experiment is getting us to is thinking about the antigen that is involved with T cells. So if you remember when we talked about B cells and antibodies, I gave you a little introduction about antibodies. Then we took a slight diversion and talked about antigens for a while. And then we went back to antibodies. That's basically what we're going to do here. So I just gave you a little intro about T cells, the T cell receptor. Now we're going to talk about the antigen for a bit, and then we're going to go back to thinking about T cells. The trick is that the antigen here is going to take me multiple days to talk about instead of part of class. And so this is going to introduce you to sort of what's up with this antigen. This is an experiment done by Zinkernagel and Doherty. They won the Nobel Prize in 1996 for this experiment. And this experiment, like honestly, pretty much every other Nobel Prize winning immunology experiment, did involve them lucking out a little bit in terms of like things they chose. If you remember with Von Behring and Kitasato, I mentioned they happened to choose the right bacteria that gave them really clean answers. Here, they actually happened to choose the right mice that gave them really clean answers. Later in the semester, we're going to actually have to deal with that with this experiment, but we're not going to deal with it now. So what I will just say is that, yes, I am oversimplifying some things now that I will have to clear up later in the semester, but right now, go with this. And so in this experiment, one of the things that we're going to have to think about is that they were using mice of two different strains. And the only thing that you really need to know about these mice of the two different strains is that every mouse of strain A is genetically identical and known. So you can think of every mouse of strain A as like infinite twins of each other. And every mouse of strain B has some different genetics, but all B's are identical. All A's are identical. A and B is not the same. And what they did in this experiment was they took a mouse of strain A and they infected that mouse with a virus. Specifically that virus is called LCMV, so that's not super important for us. And then they waited for about a week in order to get T cells responding to LCMV. So they waited and let this mouse make an immune response to LCMV, specifically a T cell immune response. And then they took out the T cells from that mouse. Specifically, they were working with CD8 T cells here. I will also fully admit I did my PhD on CD8 T cells. For a lot of reasons, if I am defaulting, I'm defaulting to a CD8 T cell in my head. So yeah, they use a CD8 here, but this is also the experiment for this, so it's not like it's just because I like CD8s. And then they also set up some other cells in the lab. And these were cells that were infected with the same virus, LCMV. They either set up cells that were infected who were also from strain A, the mouse that had the same genetics, or they set up infected cells from strain B, mouse that had different genetics. And they mixed together the CD8 T cells and the infected cells in a dish. When they set that up, there was one other thing that they did with their infected cells before they put them in a dish. And you can see these cells are labeled as the target cells. They also filled these cells with a radioactive type of chromium. And so you can see that here as yellow dots. And the reason why they did that is that they wanted to measure the ability of the CD8 T cells to kill. And so what they would do is they would put these radioactive chromium targets with CD8 T cells. And if the CD8 T cells killed, then the chromium would leak out into the media, into the supernatant, like you isolated yesterday. And it would be really easy to tell if the cells got killed or not, because you would just look, did chromium leak or not? You could basically just is there radioactivity in the supernatant or not? And that was all you had to do. It's a very, very easy assay. Well, it's actually not that easy. I've done it, but relatively straightforward. And so the goal that they wanted to see was whether or not these CD8 T cells could kill these target cells. And this is the outcome. This shows, 3 different outcomes. I'm actually going to describe a 4th outcome to you as well, or for you as well. And so, in every case, they have these purple CD8 T cells that came from a mouse of strain A. If they used target cells from strain A, that had been infected with this virus, LCMV. And remember, my other original mouse also got this same virus, LCMV. So we have a match in terms of the strain of mouse genetics, and we have a match in terms of the virus. Now the T cells worked. The T cells killed the target cells. The T cells recognized. But if they use those strain A T cells, actually we'll start with this one first. We use those strain A T cells, and we use target cells that were not infected with LCMV. So these are target cells that don't have this virus, which is the virus that these T cells were activated against. Now, the T cells do not kill the cells. So somehow the T cells know if you were infected with that virus or not. You don't want to kill the uninfected cells, which is true because we want to have ourselves live. And so if you don't have the infection, the T cells can't recognize you. What's not shown here is that you could also do this experiment where you use T cells that were generated and activated against LCMV, and you could use target cells that were infected with a different virus, like influenza. And if you did that, the T cells don't kill. The T cells only kill if they see a match in terms of the virus that the target cell has. So they are, as we saw with other adaptive immune responses, really specific. They only kill target cells that have been infected with the virus that they were activated against. So all of that's pretty standard with stuff that we've been talking about. The place where things got surprising was in this final situation, where they took their strain A mouse T cells that had been generated against LCMV, and they put them in a dish with LCMV infected cells, so infected with the same virus, but those cells are from a mouse with different genetics, strain B. And when they did that, the T cells didn't work. And this told them that T cells are looking for two things. They're looking for, do you have the right antigen? But also something about, is your cell genetically identical to mine? Is this my cell? And so they saw that T cells had two things they needed. They needed both the antigen, but also this other thing called MHC. And sometimes we refer to the fact that T cells need to see MHC, and they need to see a particular type of MHC, your MHC, not somebody else's MHC, as MHC restriction. They are restricted to act only against to only work when it's your MHC. And note that this is different than what we saw with antibodies and B cells. If you make antibodies against some microbe, and then we inject your antibodies into me, your antibodies are going to work in me. Your B cells are going to work in me. Your T cells aren't. Your T cells are only going to work for your genetics. And so what was realized by Zinkernagel and Doherty when they were describing MHC restriction, and what we now know, is that the T cell receptor, and you can see a T cell here in blue with its T cell receptor in blue, is recognizing this combination or this complex of two things together. One of them, which is shown in red, is part of a peptide from the microbe. Note that I said peptide here. When we think about the antigen in the case of an antibody, it could have been a protein, a carbohydrate, a nucleic acid, or a lipid. For a T cell, it can only be a protein because we have to have a peptide here. And this additional molecule, MHC. And so really what's going on here is that we'll have some protein from a microbe that is in our cell. And so again, this is that inside thing. That is going to get broken down inside our cell. So you can see it got broken down into little pieces. And we're going to have this other protein, MHC, that is going to display those pieces on the surface of the cell. That's how the T cell can see inside of other cells. Is that we've got this special protein on our cells that's showing off the proteins that are inside so that T cells can check them out. And when you look, the T cell receptor is actually binding to both the MHC protein and to the peptide from the microbe. And in fact, the antigen for a T cell receptor is MHC plus peptide, not one of them by themselves. You can see sort of all of this from a version of your textbook here. And so a T cell is binding to both MHC plus peptide. If it sees the MHC and the peptide that it is looking for, then we get recognition, we get killing. The T cell works. Yay. If we have a different peptide, so this might be that peptide from a different microbe, even if it's the right MHC, the T cell is not going to be able to bind. The T cell does not work. Also, if we have the right peptide, something from the right microbe, but we have a different MHC, the T cell also does not work. And so our T cells are restricted to only working against our MHC. I'm not going to go super into this right now. This is a much later in the semester problem, but I'll just tell you that this issue of MHC is the thing you have to do matching of when you do transplant matching. The reason why we have problems in transplantation is if you are getting organs from someone who has different MHC than you. That kind of is where things get all messed up. But we're going to talk about MHC and transplants so much later. And so you can see this sort of process of antigen binding and MHC function displayed here again. Today, I'm going to be, for the rest of the time, telling you some things about sort of the MHC molecule and its binding to the T cell receptor, and this one other really exceptionally critical fact about MHC. On Wednesday next week, we're going to talk more about sort of the structure and function of some details of the MHC molecule itself. And then I guess it's Friday next week, we're going to talk about sort of the degradation and the process of how you get the part of the peptide onto the MHC. So that's just where we're going through this. But one of the things I've already told you here is that the antigen that is presented by the MHC, and typically we use the word antigen and antigen presentation here. So the antigen from the microbe is always a protein. It's always a peptide. So we can't have carbohydrate, lipid, nucleic acid here. The other thing that you should know about that epitope that is being presented, again, you can imagine this is my whole antigen. The piece that gets in the MHC is the epitope, because that's the piece that's going to be binding to the T cell receptor. The other thing to know about the epitope is that A, the epitope has to be a peptide. It has to be a protein. And B, the epitope is what's known as a linear epitope. That means it's a series of amino acids that are next to each other in the primary structure. They might be on the outside of the protein, they might be on the inside, but it's not like you're going to have an epitope that's these three loops that's only there when this is folded. It's going to be some line of amino acids. So, it could be like this line, but it's going to be some sequence of amino acids that could be found in the primary sequence. Because, in fact, we're going to break the antigen into parts we're only going to have sort of little lines of amino acids that can go into the MHC molecule and be presented to the T cell. You're going to see this image a lot as we talk about MHC. For some reason, this image like really works for my brain, so I go back to it often. But this sort of shows you a cross-section of an MHC molecule, a peptide, and a T cell receptor. So if you were to imagine kind of what we're looking at right here, we're looking at it from this side instead of from this side. We're just moving to look at the other side of this. And what you can see, first of all, is that the T cell receptor is making contacts with parts of the peptide and with parts of the MHC. So the T cell receptor is really seeing and binding to and needing both of them. And you can also notice that we're seeing kind of this little area in the MHC molecule where the peptide is bound, that is officially called the peptide binding cleft. Sometimes I talk about it as like the pocket or something like that. There's like this little area that fits a peptide in it. You can see this is showing you some of the different amino acids, particularly their R groups that might have different shapes or charges. And you can see that the MHC is going to make some specific contacts with some of the amino acids of the peptide, while the T cell receptor is going to make some specific contacts with other parts of the peptide. So it's really kind of all three of these things are needed, and all three of these things have to be bound together. So you're going to see, like I said, this image a bunch of times. You're going to hear me talk about either the cleft or the pocket. So you can see that here, and you can see how the MHC molecule is binding. If we look at this same thing in a sort of a slightly more complex kind of biochemical interaction, you can see if this is the same thing. Here at the bottom, you're seeing MHC all in these gray parts, and in yellow you can see the peptide. And we'll talk about like what are the domains of MHC and all that business on Wednesday. At the top, we've got our T cell receptor. It's immunoglobulin domains, and if you notice, it's got some little loops here that are actually contacting the MHC and the peptide. It's the same loops that you saw with the antibody. It's also complementarity determining regions that are part of immunoglobulin domains. Oh my gosh, they're going to come from BDJ recombination. It's a lot of the same kind of stuff that you're seeing there. Here you're seeing it in the middle in cartoon form. In this form, you are the T cell. So this whole big like gray blob here is MHC, and this little yellow thing is peptide. And these three areas, 3A, 3B, 1B, 2B, 1A, 2A, those are your three little loops. So those are the three loops, and you can see they are actually touching the MHC and the peptide and making contacts across this. You can really almost imagine this as sort of a contiguous, sort of bumpy surface of protein structure. And you can even see here, it's showing the MHC molecule in two different ways, with this gray box, which is sort of where the T cell receptor is. So we actually know even the angle that the T cell receptor will bind to that MHC with. Yes, Jonah.
When it comes to the loops on the T cell receptor, are the R groups from the peptide binding, I guess, to those loops, or is it in between the spaces, or as you described it?
Excellent question. So what I'm going to say is that some of the R groups are going to be kind of pointing up and making contacts with the T cell receptor, while others are pointing down and making contacts with the MHC. Does that answer your question for now?
Yeah.
Okay. There's sort of more complicated parts to the answer that involve things I haven't told you yet. But for what we've talked about now, that's an answer. Okay? If you remember, when I sort of first was telling you some things about T cells, one of the things that I told you about T cells is that there are multiple kinds of T cells. There are also a few different kinds of MHC. And so this is part of why it was tricky to answer your question, Jonah, because the answer actually depends on which kind of MHC we're talking about. This is showing you the genome region that encodes MHC. You can see that there are three different regions, the class 1 region, a class 2 region, and a class 3 region. So one thing that's super interesting about this is that this whole region, this is actually the version from your textbook at the top tries to tell you how long this is in your genome. It's actually a huge region of your genome. This is the entire short arm of chromosome 6. So it's basically an entire half of one of your chromosomes that makes up this whole region. One of the ways that I can always remember that it's the short arm of chromosome 6 is that Michael Crichton wrote a book called Chromosome 6. And in the book, the idea is that they were trying to make chimps to have organs that could be transplanted into humans, and they knew MHC was important for transplantation, so they gave them the human part of chromosome 6, but it also gave them sentience. So it was like a whole crazy thing. It's quite the book. It's I'm not going to say that it's the most sane book, but that's fine. But it always is how I made it really easy for me to remember that it was chromosome 6. So we've got these three regions. One region is known as the MHC class 1 region, and it contains some MHC proteins that are known as the MHC class 1 proteins. Their names you can see here, in the case of humans, are HLA-A, HLA-B, and HLA-C. The nomenclature of the genetics will be also a big part of stuff we think about on Wednesday, next week. The MHC class 2 region has genes that are known as the MHC class 2 genes, and thus encode the MHC class 2 proteins. Class 1 encoded the class 1 genes, or had the class 1 genes which encoded the class 1 proteins. So you can see the class 2 proteins in humans. You can see their HLA-DP, HLA-DQ, HLA-DR. One of the ways I like to remember this is that class 1s have one letter, A, B, or C, and class 2s have two letters, DP, DQ, DR. There is also a class 3 region. The class 3 region of the MHC part of your genome does not contain any MHC genes at all. But it turns out that section of your genome is full of immune genes. It's like this super immunologically important area of the genome. So you can see complement is all encoded there. A bunch of cytokines are encoded there. And so it just so happens that the region between MHC class 1 and MHC class 2 encodes a lot of things that we as immunologists care a lot about. So let's imagine for a second that I was going to show you, we were going to think about, I'm going to show you a cell, and we were going to think about actin genes in that cell. Do you think that the actin genes that you got from your mom and your dad are, well, actually, I'm going to come back to that one. We're coming back to that. Don't worry. Before I do that, I'll show it to you this way. So here you can see chromosome 6 from mom and chromosome 6 from dad. You can see that there are 3 different MHC class 1s listed from mom, A1, B1, C1, A2, B2, C2 from dad. You can see 3 different ones listed from mom, 3 different ones listed from dad for class 2. And what you should notice here is that these MHC genes are codominantly expressed. So all 6 of these class 1 genes are transcribed and translated and end up on the surface of this cell. And all 6 class 2 genes are transcribed and translated on the surface of this cell. So this cell has 6 different kinds of MHC class 1 on its surface and 6 different types of MHC class 2 on its surface. What you can also notice is that mom and dad, if we just look at the MHC class 1 gene, HLA-A, mom and dad had different versions of HLA-A. Mom had HLA-A1 and dad had HLA-A2. And they make different proteins. See, like one of them's purple and one of them's green. They're totally different. If you think about the actin genes that you got from your mom and your dad, and thus the actin proteins that you got that would be made from mom and dad, what do you think you would see? Do you think you would see 2 totally different actin proteins from your mom and your dad, or do you think they'd be the same? At least based on probability. You think it would be the same. Why do you think it would be the same? They're serving the same function, and honestly, if you had got some mutations in actin and your actin got changed, like your cells wouldn't work. It's really important for us in the population of organisms that we all have the same protein that looks the same. So mom's version, dad's version, probably the same. Probably both making the same old actin protein. In this image, I'm showing you mom and dad have different A, and they have different B's, and they have different C's, and they have different DRs and DPs and DQs. And so you might say, well, what's the chance of that? Like, did she pick some super weird situation here where mom and dad have such totally different MHC genes? The answer is actually no, I didn't. That, perhaps one of the most important things that we need to understand about MHC is that the MHC genes are the most polymorphic genes in the human genome. So, to sort of rephrase that, if I went through everybody at Drew and I sequenced actin genes from mom and dad of every single student at Drew, I'd probably find like one version of actin. Everybody would mostly have the same actin, right? We would say that there was one allele of actin in the human population. I actually don't know how many alleles of actin there are, but it's a pretty small number. Most genes, we all want to have the same of that gene, but this gene is unique. In this gene, everybody's version is different. So for example, for HLA-A, and this is from your textbook, we are discovering new ones all the time, so no matter what I show you, the number's out of date. But when this was done, there were 2,980 versions of HLA-A among the human population. So I showed you mom and dad had 1 and 2. Well, there's 1, 2, 3, 4, all the way to 2,980 as the choices. For HLA-B, 3,700. For HLA-C, 2,661. For DR, we're around 1,800. For DQ, you can see there's about 800. For DP, there's about 700. And these are the most diverse genes in the human genome. Of every gene in the human genome, this is the one where there's the largest number of alleles, or the most versions. So this is the one where we're going to see variation, the most where we don't see variation in any other genes. And when we think about this, you might say, okay, HLA-A, B, and C are kind of all drawn pretty structurally similar here. How are A, B, and C different? And how is A1 different than A1712? Like, where are all the differences in these MHC class 1 genes? And when we actually look, we find that the differences between different MHC molecules, whether we're talking about class 1 or whether we're talking about class 2, are all in the pocket, are all in the peptide binding cleft. And so you can see the differences are really all in these areas that are shown in red. Everywhere else is pretty much the same structurally. It's just that we have sort of these unique pockets between each individual person's MHCs. On the left, you're actually seeing kind of the specific amino acids where we tend to see variation. And so what that means is that every person's MHCs, in fact, all 6 of your class 1s and 6 of your class 2s, would look slightly different if I drew an image like this of them, in that they would all have slightly different pockets. So this one has a little ovally thing at like the 2nd amino acid and a little cone at like the 6th. But maybe if I showed you somebody else's pocket, instead, down here in the MHC part, it would have a space for a diamond at position 4 and would bind to a different kind of peptide. So everybody's MHC is different, specifically in the pocket, which means everybody's MHC is going to be binding to a different peptide. And so here is kind of one way of thinking about this. And I'm also going to draw this for a second. See how poorly I can draw this. All right, so let's imagine that this is my pathogen protein. It has a whole bunch of amino acids, and we're going to unfold the whole protein into a line. And so part of it is the pink part, and then there's some green part. I need some more colors. Then there's some blue part, and there's some purple part, there's some orange. So there's my peptide. You could just imagine we could break it up into pieces. It's really one long string of amino acids. In the slide, you're seeing single letter abbreviations of amino acids. They're trying to show you different parts. And maybe we have five people. So we have David, we have Brenna, we have Shelby, we have Brooke, and we have Brian. Sorry, I only had 5. It turns out that David's cells might have MHC with a green pocket. So it can only fit green peptides. Brenna might have MHC with an orange pocket. So Brenna is only going to be able to have her MHC fit this peptide. Shelby, red pocket. Brooke has the purple pocket. And I don't even know what color I didn't do yet. Blue. Brian has the blue pocket. So each person is going to show off on the surface of their infected cell a different part of this pathogen's peptide. And it's going to happen that every pathogen, if we think about all the proteins in the whole pathogen, somewhere it's going to have a green part. And so somewhere it's going to have a piece that will fit into David's MHC pocket. Somewhere in a whole pathogen, out of all of its many proteins, there's going to be an orange part. And so some part is going to be able to be presented on Brenna's MHC. But everybody is going to be presenting a slightly different part of each pathogen. So what this is trying to show you on this slide is this is one kind of MHC that it's listing here as K of B. And this is showing you there are different peptides from a bunch of different microbes that can all be shown by that kind of MHC. while this other kind of MHC has some separate types of peptides that can be shown. But like they both have some part of influenza they can show. And the thing that is in common with all of the peptides that a particular MHC shows, like here I'm saying the thing that all pink peptides have in common, for example, is perhaps that they all have leucine at position 8. So they can all fit into a pocket that has a spot for leucine at around position 8. And these all have phenylalanine or tyrosine at position 5. While in this pocket, you need to be able to fit this pocket, you need a tyrosine at position 2 and a valine or isoleucine at position 9. And so we have this variation in everybody's MHC pocket. And so everybody's presenting something from each microbe, but it's going to be a different something from each microbe. Do you have a question, Brenna?
I don't know how to word my question.
Okay.
But so does everyone have their own pocket? Like there's just one type of pocket that we have that works for everything? Or do we have multiple different types of pockets? Because what if that one pocket doesn't?
So you have 6 class 1 pockets and 6 class 2 pockets on your cells. And so the hope is that with your 6 different class 1 pockets, for this person, the 6 are A1, A2, B1, B2, C1, C2. They've got 2 A's, 2 B's, and 2 C's. The hope is that for every microbe, you can get at least one of your 6 can get something. And generally all 6 of them can. So does that make sense for you?
For some reason I was thinking we just had one pocket.
No. You have 2 A's, one from mom, one from dad, 2 B's, one from mom, one from dad, 2 C's, one from mom, one from dad. And the thing that's really important about this is now let's imagine we've got T cells that are going to recognize our 5 infected cells. These T cells are recognizing different epitopes of the pathogen. David's T cell is going to recognize the green epitope from this pathogen. Brenna's is recognizing the orange epitope. Shelby's is recognizing the red. Brooks is recognizing the purple. Brian's is recognizing the blue. So in fact, each person's T cell response is going to be different because they have different MHCs showing off different peptides. It turns out that some epitopes are better than others in terms of targeting a pathogen. So let's imagine for a second that the blue T cell is really, really, really good at killing this pathogen or dealing with this pathogen. And the orange T cell is terrible at killing this pathogen. In that case, when Brian gets this pathogen, he's not going to get sick because his T cells are going to kill it super well. While Brenna is going to get really sick because her T cells aren't going to work super well against this pathogen. So because of this variation in MHC types, we're going to end up with variation in how well we control any particular microbe. And so you can see this, here. So this is just one example. So this is showing a bunch of different MHC types and what happens to people who have HIV with each of these. So sometimes you get slow disease, sometimes you get rapid disease, sometimes you have a lot of virus, sometimes you have a little bit of virus. And it's based on whether or not you have the sort of quote unquote good MHC that will help you deal with that microbe. You might look at this, and again, to sort of address Brenna's question, you got 6 chances to have a good class 1, you got 6 chances to have a good class 2. And the selection pressures on this end up working out differently than other genes because we have this selection to have as many different variations of MHC as possible amongst the human population. So if you get a mutation in your MHC, it's like, woohoo, new version, instead of, oh, that's bad, you're gonna die. Because if you look at this, HIV infection one, for example, one of the most famous associations with HIV and HLA is with HLA-B27. If you have HLA-B27 and you get HIV, you are not really going to get particularly sick at all. You're going to have incredibly low amounts of virus. Your body's going to fight that virus really well. So you might be like, I would like HLA-B27, please. Why is it that everyone in the world does not have HLA-B27? Well, HLA-B27 is great for HIV, but there are some other pathogens where HLA-B27 is terrible. And it's the one that results in death. And so for each individual person, your HLAs might be good or bad. For whatever pandemic happens, you might be one of the lucky ones or you might be one of the non-lucky ones. But for us as a human population, because we have such diverse HLA molecules, there's no pathogen that can kill us all. Somebody has the right MHC combo to deal with whatever pathogen. Somebody has the right set of pockets to help them deal with that pathogen. Even though it might not be great for you as an individual, for us as a human population, it's great that we have this diversity. And this is why we've had this selection for this diversity. And so just to sort of point out a couple of examples, MHC type is also very much related to whether or not you have autoimmune disease. And so, for example, B27 that I told you is so good for HIV, people with B27 are also very likely to get a particular autoimmune disease called ankylosing spondylitis. I am possibly getting diagnosed with an autoimmune disease at one of my doctor's appointments I have next week, and I'm guessing that what they're going to do is they're going to check my HLA type. In fact, it's like there are so many diseases where people have done genetic studies to find out what gene is most important, and they come up with MHC. And in fact, sometimes when I see that, I'm like, thank you, Captain Obvious, that this immune disease is related to MHC. Like, you just spent how many $1 million to find out that your immune disease is an immune disease? But anyway, that's my snarky answer. One other thing that's really interesting is that if we actually look around the world, the frequencies of different MHC types seem to vary. So there are some MHC types are particularly common in some populations and are particularly rare in some populations. A lot of this seems to be a genetic drift thing, though we're not totally sure. Anthropologists actually use MHC to do things like when they're sort of arguing about, well, which line of descent did this group of people have, they figure out whose MHC they're most closely related to. And so so there's a massive diversity of MHC around the world. And so we can end up today talking about a couple of sort of MHC fun fact things. One of them is completely ridiculous, but it's like, there's literature about it. It's just whatever, it's craziness. So if you look at this, you got mom's MHC, you got dad's MHC. Do you hope, if you went home now and tried to look at mom and dad's MHCs, do you hope mom and dad have similar MHCs or different ones? Different. You hope they have different ones so they could give you different ones, right? Like if mom and dad had the same MHC and gave you the same MHC, then you don't have so many pocket choices. That would be bad, right? And you can imagine it might be good for us that if we generally reproduced with people who had different MHCs than us. There is actually a study that is nuts. It's just nuts. Where they tried to see whether people found others with similar or different MHCs more attractive. I don't know how supposedly we're sensing other people's MHC. I can come up with a weird way through microbiomes, but I don't know. But this study has been done over and over and over again, where they had male and female college students where they figured out their MHC types. They had the male college students work out in a t-shirt, and then they gave the t-shirts to the women and asked them which one smelled the best, which one smelled the most attractive. And apparently you are most attracted to people who have the most diverse MHCs from you are. So sometime if you are like, I don't know what it is about that person, maybe it's their MHC. But we can also think a little bit about what would happen to a population if we didn't have this MHC diversity. And so there are some organisms that we know about who don't have MHC diversity. In both cases that I'm going to tell you about of organisms who don't have MHC diversity, what we think happened is that there was some other event that happened to that organism that killed most of the members of that population, and so there were like 3 left. And so all of those animals who are currently present are the progeny of those 3. So like they just don't have very much diversity in MHC because they all have the same MHC from those original 3. So the idea is that there was some kind of population bottleneck, and thus that organism is really has very, very similar MHCs among all members of the population. What might you hypothesize would be something that would happen with one of those organisms? There's 2 big things that we see. One of them you can hypothesize, one of them you probably can't. So what might you hypothesize as happening to that population of organisms? Yeah, David.
They're really susceptible to some kind of some specific microbe or pathogen.
One issue is that those organisms might be particularly susceptible to some microorganism or some pathogen. And so it's possible that there is some microbe that could kill them all because they don't have enough diversity in MHC to deal with this. So these are some pictures from my short trek in South Africa, and we go to cheetah rescue at one point while we're there, and one of the things we have to do is do a lot of disinfection of our hands and feet and anything before we go near the cheetahs, because cheetahs are actually one of the 2 organisms we know that have almost no MHC diversity, and so cheetahs are particularly susceptible to different pathogens. And it is in fact possible the cheetah population is endangered, and one of the big reasons the cheetahs are endangered is because there's so little MHC diversity and they have outbreaks potentially.
And there's no way to resolve that.
Other than like many generations of picking up mutations and protecting the cheetahs. I know I'm at 12:55, but I have to tell you about the one other one because it's just like, it's really fun. So the other organism that is known for its non-diverse MHC is the Tasmanian devil. This is a Tasmanian devil. I really like this picture of the Tasmanian devil. If you go to the Tasmanian devil's Wikipedia page, one of the things you learn about Tasmanian devils is how much of their life involves biting, particularly biting each other in the face. And so I like this picture because it looks like this Tasmanian devil is ready to bite. So let's imagine I had some disgusting skin cancer coming out of my hand right here on my skin. And then let's imagine I wiped it on David. Would David get cancer? No. And you're like, no, of course not. That's not how cancer works. The actual reason why David wouldn't get cancer is because our MHCs are different. And his immune system would actually reject my cancer. Tasmanian devils don't have MHC diversity. Almost none. They have very, very little. Tasmanian devils are probably going to go extinct because there is a face tumor in the Tasmanian devil population called Tasmanian devil transmissible tumor that they actually transmit from one to another when they bite each other in the face. And because they have no MHC diversity, they cannot reject this tumor. And so, like I said, these organisms are probably going to go extinct because as they are biting each other, they're spreading this tumor, and they cannot reject it because of this lack of MHC diversity. There is actually, this is one of a very small number of transmissible tumors known because it's only possible when you have this lack of MHC diversity. In fact, there's 2 in Tasmanian devils. There's also one in a genital tumor in some feral dogs in South America. And so while you are having an awesome break, be thankful for your MHC diversity and all the ways that your MHC is helping you out. And I will see you guys after break when we will start talking more about fun and exciting MHC stuff.